Hi, everybody, and welcome to today's lesson. Today, we are going to be focusing on animal breeding and genetics. So let's dive right in. So genetics is the study of what makes us who we are. And I'm not talking about your personality. I'm talking about, well, I mean, to an extent, your personality. Essentially everything that makes up existence. Genetics is the foundation of life in that for an animal to be what it is or what it could become, those genes from its parents need to be passed on from one generation to the next, and then that individual needs to be able to pass along its genetic information. And if these animals or people cannot do that or fail to do that, that means life as we know it does not exist. So genetics is the study of essentially the code that makes an animal or a person who they are. And genetics is important to livestock because we have certain things that we want our animals to be able to do, right? So if we have dairy cows, we want them to be able to produce enough milk, right? If we're raising beef cattle, we want them to produce, muscle, but we also need them to be able to have babies every year and raise their babies. Sheep, might grow wool, they might grow meat. Goats could grow meat or mohair or, be milking goats. So we have these things that we expect our animals to do or that we ask our animals to do. And if you have an animal that, for the past couple of years has, struggled to give birth or hasn't been a great mother or, any number of problems, you're not going to want to keep breeding those bad things, right? Because odds are if you have an animal that's a bad mother, she's going to teach her daughters the same thing. And so her daughters are not going to be great mothers either. And you're going to perpetuate this bad thing over and over, right? So instead of doing that, we pick the mothers that are good and we breed them, and then they teach their daughters to be good mothers in theory. And this trait gets passed on from generation to generation. And while I'm talking about mothers teaching their daughters, a lot of it is instinct, and instinct is genetically related. So when we have these animals that have traits or things that we like, we consider positive, we want to be able to perpetuate or pass these things on. And that is genetics. So how does genetic information get transmitted? Well, it gets transmitted during cell replication. So as you may or may not know, cells don't last forever. Cells exist for typically a relatively short period of time. It depends on the type of cell. But those cells eventually replicate and the original cells will die over time. And so we have two different processes of genetic transmission. We have meiosis and then we have mitosis. Now, when we're talking about gametes, so like the eggs or the sperm cells, those replicate typically through meiosis. And then all of the other cells in the body replicate via mitosis. Mitosis is like an exact copy. So if you took a document and you made a copy of it, it's the exact same document, and there's two, right? Meiosis involves some crossover and mixing up of genetic information. So you're not getting an exact cookie cutter copy. You're getting a mixture, a cocktail of different genetic makeups, which is why you might have a sibling, a brother or a sister, who's not a twin, that still looks relatively similar, but not exactly the same, right? Do any of you have more of a resemblance to your mother versus your father? I look more like my father than I do like my mother, and my brother looks much more like our mother. So even though we are genetically related, because of this meiosis and meiotic transmission, we don't all look exactly the same. And that's really, really important for genetic diversity. So let's talk a little bit about cells. So you can think of cells kind of like building blocks of organisms. And within the cell is essentially a little factory. And, it contains little, what we call organelles. So they're not organs in the sense of a heart or a brain is an organ, but they do have these structures that perform very similar functions to an organ, but they're not technically divided, the same. They're not multicellular. So we have the nucleus of the cell, which is where all of the DNA replication is happening. And then we have the Golgi body, which is responsible for packaging and shipping. The smooth endoplasmic reticulum is SER, smooth endoplasmic reticulum, and the endoplasmic reticulum are also responsible for manufacturing and shipping different nutrients. And then ribosomes are little tiny kind of shipping containers that move around the cell and usually take things outside of the cell. Mitochondria, you probably have heard, are the powerhouse of the cell. This is where all of the cellular energy is going to be produced here in the mitochondria. Fun fact about mitochondria. Mitochondria are inherited from your mother. So you do not inherit mitochondria from your father. You inherit it strictly from your mother. And so if you are looking for parental matches or familial relations, you can actually look at the mitochondria and match them up to females in a family because they will all be very, very similar. So fun fact about mitochondria, I studied mitochondria for my PhD, and they're very, very fascinating. Another fun fact about mitochondria is that in cattle, and only as far as we know in cattle, they have a very special kind of mitochondria in their oocytes. So in the eggs that the female cow makes, they have a very unique type of mitochondria. It's called a hooded mitochondria, and they have a little hook on the end of them. And we don't know why, we don't know what they're for, but cattle are the only ones that we know of right now that have this hooded mitochondria. I don't know why you would need to know that, but now you do, and you're never going to unknow it. You're welcome. All right, moving on. So the nucleus of the cell, like I said, is where all of the genetic replication is going to be taking place. And specifically, those are contained, all that genetic information is contained in these chromosomes. So you can kind of see in this picture here, this picture is showing meiosis, the process of meiosis, which if you've ever taken a biology course, you probably have heard of meiosis and mitosis before. And this is just to kind of give you a little bit of a refresher about meiosis and mitosis. So you start off with a single cell, and then there is, chromosomes that exist in these cells, and those chromosomes condense and pair up together, and then there's a certain amount of crossover that's taking place. You can kind of see here in this corner, some of the chromosome on the red side is touching the blue side, and some of the blue side is touching the red side, and so they share that genetic material, and now they replicate. That cell divides, and then you have another round of divisions so that those sister chromatids are broken apart, and you eventually wind up with 4 single chromosome cells, and they have crossed over some of their genetic information. So each one of these cells is just a little bit different. And so if these turn into eggs, each one of these eggs is going to have a little bit of different genetic information from whoever parent this is. So if this is the dam, we've got this one, and then if we get a sperm from the sire, we could potentially get this one, but there are a lot of different combinations here, right? So this is why offspring can look different from year to year. So you may never get, even if you use the same pairing, you breed the same bull to the same cow, she might have an excellent calf one year and an okay calf the next year. And that is because of this genetic diversity that exists. Okay, so when we're talking about the difference between meiosis and mitosis, mitosis actually reduces the number of chromosomes that are present in the cell by a half. So you can see we start off here with a whole chromosome, and then they pair up together, and then eventually at the end you get essentially half of what you started off with. that make sense? So you get half of what you started off with because they pair up right here and then you get half. Okay, and then in mitosis, there is no reduction. It's the exact same number of chromosomes, all right? Meiosis only occurs in gamete cells. Gametes are sperm and eggs. So that is the only place you see meiosis. All of the other cells have mitotic division, because if you think about it, if you have skin cells and they replicated via meiosis, you might have all sorts of different kinds of skin cells. You never know what you're going to get. That's not going to be great if we're just trying to replace dead skin cells, right? So we need essentially a cookie cutter of the same type of skin cells. So that's why mitosis works better in all of the other cells in the body. But for our procreation cells, so the eggs and the sperm, we need that genetic diversity to make sure that things don't hiccup along the way. All right, so everything that dictates the way an animal or a person looked is contained in what we call their DNA. And if you've ever heard the term, it stands for deoxyribonucleic acid. That's what DNA stands for. And it's essentially a series of nucleic acids that dictate what genes are turned on and what genes are turned off. And those genes code for specific traits or functions in the body. And within DNA, well, DNA itself is kind of a long strand of this code, and it's really, really, really long. And so to fit it into a cell, it's actually condensed, really, really tiny little coils, and those coils are put together in what we call chromosomes. Okay, so those chromosomes contain different what we call genes, and genes are a fancy word, I guess, for a trait. What I'm doing right now is looking up how long a DNA strand is. So I wanted to see if you pulled it apart. Let's see. So if you stretched the DNA in one cell all the way out, it says it would be about 2 meters long. And the DNA in all of your cells put together would be twice the diameter of the solar system. So this is from sciencefocus.com, but I knew that DNA was really long and I wanted to give you something to compare it to. So this means that all the DNA in all of your cells would be twice the diameter of a solar system. That is a really long. And if you think about how many cells are in the body and how much DNA is packed into each cell, there's enough DNA in each cell to essentially re-replicate a person. But because there are certain genes that are turned on and off in each cell, that's why that's not possible. Only specific types of cells can essentially replicate an entire organism. One of those is embryonic cells, embryonic stem cells. So you have these, coils of DNA that combine into chromosomes. And then within these chromosomes are little segments, and these segments code for what we call genes. Okay, so this is kind of a better illustration, I feel. So this right here is a chromosome, and this chromosome contains just coils and coils and coils of DNA. And then these little specific bands here are code for a specific gene. So they're still DNA, but this section of DNA codes for a specific protein. Okay, and those proteins will then go on and regulate some part of the body. So they might be what make your eye color the way it is. They might dictate what color your hair is, how thick your fingernails are, what color your body hair is. All of that is part of the genetic makeup of an organism. And those genetic makeups are proteins. So when we talk about animal breeding, we're not talking about reproduction here. We are talking about decisions. We're talking about which animals we are going to be using to procreate the species. So it's not the same as discussing reproduction. We're not talking about eggs and sperm and reproductive parts. We're talking about traits and what we want to continue or propagate. So we're talking about selection. So animal selection includes making decisions about which animals we will use. So if you have a bull and he is really fine boned and he's really thin and he's tiny and he never grew well, he's probably not one that you're going to want to breed over and over. If you have a cow and this cow had produced one calf and then she didn't get pregnant and then she did produce a calf the next year, she's just not one that we want to keep going with, right? So you want to make decisions for good animals. If you have a dairy cow and she is your top producing dairy cow, and you have a bull and his daughters tend to produce really, really high milking animals, those two together might make a really good pair, right? So you're picking which animals will be used for breeding and then how long they're going to be a part of our breeding stock. Now, if you're talking about commercial production for like beef animals or meat animals in general, generally we'll let them be a part of our herd until they can't produce for us anymore. So it's very unusual for a producer to be like, yes, I will keep this cow for 5 years and then I'm going to get rid of her. We have cows in our herd at Shasta College right now that are well over 10 years old. And as long as they're still doing well, they're good moms, they're having babies, they seem healthy, then we are planning to keep those cows in our herd. So, but that is how we have made our decision. Some producers might say, well, I want to refresh my stock every so often, so my animals will be a part of this herd for X number of years, and then they will be rotated out. Now, with our breeding males, we do tend to rotate them out a little more often than our females. And that is because after a while, if we have a buck and we breed him to all of our does, and then we keep some of those daughters that are born, after a while, we're not going to have any females that can be bred back to this buck, right? Because we can't breed his daughters back to him. So we have to have a different set of genetics to breed those daughters to. So typically we will rotate out our boys every couple of years and we'll bring a new one in behind them to just keep that genetic diversity up. All right. So you can make decisions based on how long they'll be a part of the stock and how many progeny we'll let them create. So some people will be like, well, we'll let him, father a couple rounds of calves for us and then he's going to be gone. So that's how you're making decisions based on selection. When you're talking about mating, you're talking about which female mates specifically with which male. All right. So selection is picking the animals we're using for breeding. Mating is deciding which pairs will go together or which bull will be bred to which cow. So this is easier if you're artificially inseminating because you can pick from a larger selection of males. When we artificially inseminated our cows, we had 4 or 5 bulls worth of semen that we used, and we went through and we picked which bull we thought would pair best with which cow, and that's how we did that. What we're trying to do here is we're trying to breed better offspring. We want to make the best animals possible, and so to do that we need to kind of figure out which traits are able to be passed on. So we have this range of what we call heritability, and that's the ability for an animal to pass on a specific trait. Some traits are very heritable, like temperament, and some are not that heritable, like fertility. So when we're breeding an animal, we don't just go out and look physically at that animal and be like, yep, that's a really attractive looking bull and that's a really attractive looking cow, so let's go ahead and put them together. It's based on more of that than that. It's not just based on their looks, because you can have an animal that looks fantastic, but when you breed that animal, it never throws fantastic offspring. Okay, so it's about more than just how they look. It's about their genetic background. So we call that their genetic background, we call that their genotype, and that's their genetic makeup. That is the blueprint for that animal. And then we have a phenotype, which is the outside, what the animal actually looks like when you look at it with your eyes. Okay, so either of these things is not a fail-safe. You can't just look at an animal and be like, yep, that animal is going to throw beautiful babies. And you also can't just look at a pedigree and be like, look at this pedigree. This is the most impressive pedigree I've ever seen. This animal is going to throw beautiful babies. You have to work with both of them together, because an animal can have a very impressive genetic background or pedigree and still not produce great offspring. And on the flip side, they could look fantastic, but not be able to pass on those genes. So these two things have to work together. And so when we're talking about genetic principles and where things are located as far as the gene and the chromosomes and stuff, we're talking about, if we're talking about the actual physical location of a specific gene, we're talking about the gene's locus. Okay, so this is the region of the chromosome that contains the specific gene. So if we're talking about coat color, where in the chromosome the coat color gene is physically, that is the gene's locus. Okay, and then we have different coat colors, right? So we can have black coat colors, we can have red ones, gray ones, roan colored, speckled, striped. There's all sorts of different colors out there, right? So the gene itself is for coat color, right? But the different variations of that gene, so red versus black versus white versus striped, those are called alleles. It is one of the alternative forms of a gene. So if you have red hair, then the locus of the gene is somewhere in your DNA, right? And the gene itself is hair color. And the allele, or the version of hair color that you have, is red. And maybe your parents have a different allele. Maybe they have brown hair or blonde hair. So the allele is the alternative form, or forms that are available at a locus, or the different variations of a gene. The gene is still coat color, but there are different alleles for the gene. Okay, and this is possible because of that meiotic division. Okay, so because you're getting a copy of a gene from your mom and a copy of a gene from your dad, and when those, sperm and eggs, when they divided, when everything was being made, there was crossover, there was, different genetic materials shuffling around. And so that is one of the positive benefits of meiotic division. So if we get an animal that's exhibiting a trait, say black coat color, what is happening during this time? What's happening is that the animal has the gene for black coat color turned on, and it has other genes. So maybe it got a copy of black coat color from its dad, and it got a copy of the red coat color from its mom. And what we're seeing is a black calf. All right, that means that that calf has the genes for black coat color turned on. And even though it might have gotten a copy of the red coat color from mom, that gene, that allele, is not turned on. Okay, so when an animal has certain genes that are turned on or off, we call that genetic expression. So things like coat color, if the animal has horns, the fat content, the muscle content, and the bone, these are all forms of genetic expression. And these are just some of them. Anything that is going on with the body at all is some sort of gene expression, even stuff that you can't see. All right. And so when we're talking about what you can see versus what genetically might be there but can't be seen, we're talking about dominance versus recessiveness, okay? So for a dominant allele, this is an allele that can overshadow another, all right? When we have a dominant trait, we have black, say black coat color, right? And even though that calf may have gotten a red coat color copy from its mom, the black coat color is dominant. And that means that if you have a copy of it, it's going to overshadow the other one. So if the animal is black, it has the dominant allele for black coat color. If the animal was red, then it would not have the black dominant coat color. Because if that dominant color is present, that means the animal cannot express the what we call recessive color. So a recessive allele is one that is only expressed if there's no dominant allele present, okay? So this means that if there is no copy of the dominant allele, so in this case, the black coat color, then that means the animal can express the red coat color, all right? So, for example, if you had a red cow and a red bull and the calf was red, that's because there's no copy of this black one in there at all. But if you breed a red cow to a black bull, that calf will be black, with exceptions. There's some exceptions, but this is generally how it works, all right? So, we carry two copies of a gene, two alleles from a gene. We get one from mom and one from dad, and they might be the same, but they very well could be different, like with the coat color thing. So if an individual carries two dominant copies, two dominant alleles for that gene, we tend to represent that in capital letters. All right, so if we had a bull that was black and a cow that was black, and we decided that the capital B was black, then the bull and the cow would be represented by a double B, capital B, okay? First letter is the bull, second letter is the cow. So this is the allele from the bull, and this is the allele from the cow, okay? Now let's talk about if we had an animal with horns versus no horns, okay? Say we got a copy of the horn gene from dad and a copy of the horn gene from mom, and we decided that the dominant gene is going to be uppercase H, H for horn, okay? So dad gave us an uppercase H because he had horns, and mom also had horns, so she gave us an uppercase H. So this is what the baby would look like, all right? One letter from dad, one letter from mom, and in this case, they're both dominant. So when they are both the same, when they have two dominant alleles, we call them homozygous dominant. Homozygous dominant. You will need to know this, so make sure that it's straight, and if it's not, you need to let me know so I can explain it a different way. All right, if we have an individual that has one dominant copy and one recessive copy, then that individual is called heterozygous. There's no dominant, there's no recessive, just heterozygous. Hetero meaning different. Homozygous, homo meaning the same, okay? Homozygous dominant means they have the same dominant allele. Heterozygous means that there's one of each, okay? So typically, the recessive allele is represented with a lowercase letter. So if we go back to our coat color example, we decided that black, the dominant color, would be indicated by an uppercase B. All right, now if we had red, which is a recessive color, that would be represented by a lowercase b. It would not be R because we've decided that the coat color trait is B, regardless of what color it is, all right? So the dominant coat color we decided is black, and that is represented by the uppercase B. So the recessive allele is automatically the lowercase letter. Same letter, just lowercase, Okay, so if we got from dad a copy of the dominant gene because he was black, and from mom, we got a copy of the recessive gene because she was red, this would be our baby. Uppercase from dad, lowercase from mom. And this individual, in this case, would have a black coat, all right, because he has a copy of the dominant gene. Okay, so if we go back to our horned example, we decided that the trait horns would be represented by H, right? And we have the uppercase H here. And so that means that animals without horns would be represented with the lowercase h. So if we had a dad with horns and a mom without, we would get one copy of the horned trait and one copy of the non-horned or polled trait. So this individual would be horned because it has one of each, and one copy of the dominant means it's expressed. All right, lastly, we have the homozygous recessive trait. So this is an individual that carries two recessive alleles for the gene, okay? So if dad was red and mom was red, this baby would be red because it's got two copies, no dominant alleles, only recessive ones, okay? And then with the horned example, if dad didn't have horns and mom didn't have horns, baby is not going to have horns in this example. Does that make sense? I would pause and make sure this really sinks in because it's easy to get it confused. A lot of students get it confused. Go back and review this little section until it makes sense because this is very important, and all of the other stuff following this is not going to make as much sense if this doesn't make sense to you. Okay? So when we talk about genetic selection, we have different types of traits, okay? So we have some traits that we call polygenic, and that means that to change this trait, then we have to change a lot of genes because typically a trait, so things like fertility or feed efficiency or disease susceptibility, those things don't just have one gene affiliated with them. There are many genes that factor into how fertile an animal is. They don't just have a fertility gene, okay? And so that means that it's much harder to change these traits because you have to change multiple genes along with it. Does that make sense? So a lot of these traits are often our more economically relevant traits. So things like fertility, rate of gain, those are really important, right? Heat tolerance, longevity, these are all things that make a big difference to us, and they are really hard to change because you can't just change all of these genes at once, and in some instances we don't even know all of the genes that influence this particular trait. Oftentimes we do, but doesn't make it any harder to change them. And so when many genes influence a trait, we call that polygenic. Poly meaning many and genic meaning genes, right? So many genes influence this particular trait. We also have traits called simply inherited traits, which are affected by a few genes or even one specific gene. Okay, so these are much easier to change, these simply inherited traits. Things like coat color, an animal being horned or polled, which means polled means they don't have horns, and the animal's eye color. These are all things that potentially are only affected by a few genes, much easier to change. All right, if we are talking about the entire genetic makeup of an animal, we are talking about the genome of an animal. These are all of the genes, all of the alleles, and all of the locations of said genes, all being identified. The genome of an animal depends, I mean, if we have a genome map for an animal, it depends on the species. So sheep are done, cattle are done, and I'm pretty sure that pigs are done. So for a pig, for a sheep, we know all of the different variations of a gene and where those genes are located on the chromosome. It's pretty amazing. If you look up a genome map of a sheep, you can actually, let's see, I'm gonna open it up right here. Genome map sheep. You can click on this first thing here, US sheep genome here, and then here we go. Ovis Aries 3.1 is available. So you can click on it and it'll actually show you the chromosomes, the gene bank sequence, and the reference sequence. And it's kind of confusing if you look at it, but the point here is that it's been done and it's taken millions of dollars and a lot of time to map out the genome sequences. Why is this important? Well, it's important because if we know where the genes are and the variations of the genes, it's easier for us to select specifically for different changes that we'd like to make in the animal. And I'm not talking about genetic modification by inserting DNA or deleting DNA. I'm talking about selection. But on the flip side, if we are doing research and we need to, this is really important, I guess, more in mice, but if we are studying cancer in mice and we want to shut off specific genes that prevent certain types of cancer, or if we want to enhance genes that promote certain types of cancer, we can do these things and create mice that are going to get a very specific type of cancer so that we can look at treatments. Same thing with vaccines. So we have these genomes mapped because we know exactly where the genes are, and we can change them a little bit easier. Like I said, this is more important, I would say, in research animals like mice, but it's still important for things like vaccines and medications for all species. So when we talk about the size of a genome, the simpler the organism, the smaller the genome. The more complex an organism is, the larger their genome is. So let's talk a little bit about what makes good things versus bad things. All right, we're talking about changing traits and desirable characteristics. And so what exactly makes something desirable versus non-desirable? And some of the things that dictate what is good or bad falls on the producers. So if you are a producer and you have goals, like you are a dairy farm, and so you are producing milk, you have set goals, how much milk you need to produce in order to not go under, right? You have to be able to make a certain amount of profit. How much resources do you have? How much in resources? You know, do you grow your own hay? Do you have, you know, a lot of money? That would be nice, right? What is the market like in your area? If you are producing a certain product, it has to meet a market in your area, right? And if you're looking at things like the disposition of the animals, some animals can be very, very dangerous if they don't have the right disposition, right? We're talking about things like cattle that could kill you very easily. So disposition might play a role here. The consumers also play a role in what dictates good or bad. If you go to the grocery store and you want to pick out a steak, typically you're not going to pick out the largest steak there, right? You're also probably not going to pick out the smallest steak there. You're going to pick something that's pretty much right in the middle. So you as the consumer and what you purchase is going to dictate what makes a good or a bad trait. If you're breeding, if we, the producers, are breeding all of these animals with these huge ribeye steaks and nobody's buying them, that's not going to be good for us. So we need to make sure that we're paying attention to the consumers. Things like marbling, how much fat is present. The purchases that are made in the grocery store dictate how we change that. The amount of leanness that an animal has, the color of the animal, wool, fleece, that all makes a difference, and the consumers purchasing it are going to influence the market. Okay, we're going to stop there, and I'm going to record another lesson for the rest of this PowerPoint.